Okay guys, so on Google Classroom, our latest unit is just our virus unit. Okay, we have our notes attached, which I'm about to just really quick go through. This is me lecturing the notes, and here's that first Amoeba Sister video. Okay, then today we're doing a quiz review, a quiz, and a workbook lesson. Okay, if we go, just like I lectured last time, to the Lohmann review sheet that we got to use on our benchmark and look at what we learned on viruses, this is all they tell us. Structure, capsid, protein coat, nucleic acid core, DNA or RNA. So guys, the first thing we're going to see in this PowerPoint is viruses are made up of 2 biomolecules. We're made up of all 4, CHO, CHON, CHOMP, carb, lipid, protein, nucleic acids. Viruses are just the protein capsid. It needs a shell. And guys, remember, proteins build, right? They're builders. Guess what? They built that shell, and it has to have DNA or RNA. It has to have nucleic acid, okay? Surface markers, proteins for attachment. So we are going to see in the lytic and lysogenic cycle, they do have to attach, okay. Comparing to cells. First of all, we know viruses are dead, okay? So if we take antibiotics, anti meaning not, bio living, we're gonna kill the living. Are viruses living? So can antibiotics kill them? Antibiotics just kill bacteria. Viruses are dead. So if you have the flu, which is a virus, and your mom really wants that doctor to prescribe you an antibiotic, will that help you? No. Okay? But if you have strep throat from streptococcal bacteria, you have to get an antibiotic. Okay. They do have DNA like cells, but they are not cellular. They don't reproduce without a host. They need a host. They cannot metabolize, okay? And they do not have organelles like cells do. Okay, how do they spread? So remember, we do have to pick up viruses off of surfaces. In both the lytic, match, quick cycle, lysogenic, longer word, longer, sneakier cycle. The virus has to get in, attach, put its viral DNA in. Lytic gets you sick quickly, okay? Lysogenic builds, and then basically explodes whenever your immune system gets low, okay? Lytic is the quick cycle like flus and colds. Lysogenic is the longer that remains dormant, and then you have outbreaks like AIDS, HIV, herpes, okay? Also, y'all, when the little viruses bust out of the cell, what happens to the cell? It dies, okay? So you're always destroying cells when viruses are replicated and then leave the cell. Okay, that's exactly what happened in our notes. So if I will go to our notes PowerPoint, and this is going to be really quickly, just to reiterate what we already learned.
If viruses are dead, then how do they sit?
That is literally like one of those weird questions we don't know. So viruses are dead. They're literally not alive. How do they somehow get into us and make us so sick? That's just like one of those crazy.
Germs, all that.
Yeah, but if they're dead, how do they even know to attach and inject their DNA? We just don't know. Yeah, that's a really good question. So, first off, viruses, like we said, they're dead. They do not fulfill the characteristics of living things, which are they cannot reproduce on their own. They need what's called a host. They cannot metabolize and use energy. Therefore, they are dead. Antibiotics don't work on them. Okay? Size comparison. Eukaryotes, they're so organized. Doesn't that eukaryotic cell look like a little town with all of its little buildings doing its own little jobs? Okay, then we got a prokaryote, that dumb bacteria 100 times smaller. And then we got these viruses 100 more times smaller. Okay, eukaryotes and prokaryotes, they're alive. Look up here. They have all 4 biomolecules, carbs, lipids, proteins, nucleic acids. What are the two that viruses have? They have a shell made up of protein, build, and they have what inside? Nucleic acid, DNA, double helix, good. Outside protein, inside nucleic acid. Outside protein, all of these are for attaching, okay? We got 2 cycles. Lytic, lit match, fast, okay? Attach, viral DNA entry. Replicate all the DNA. We're basically tricking that host cell to make virus DNA and not host cell DNA, okay? Assembly, release, killing the host, okay? This is really quick, okay? This is like colds and flus, okay? Then you got the longer, sneakier cycle, the lysogenic, where in general, it inserts its DNA and it remains dormant, and at any time it can enter the lytic cycle. And that's why people who have AIDS, HIV, herpes have outbreaks of areas of all this cells busting out of there, or the viruses busting out of the cells.
It's fine. I have a question.
Mm-hmm.
So, if these want little answers, how are we supposed to give it little answers?
Yeah, like, are we supposed to be on that workbook lesson right now?
I'm just asking because it says what happens to the host cell in a lysogenic cycle.
Okay, and what would y'all write? What happens to the host cell at the end of the lysogenic cycle?
We're not talking about that right now.
Yeah, we're not supposed to be talking about it. Okay, so remember, lytic, quick, explode out. Lysogenic, remains dormant for a long time, but then goes to lytic whenever you have an outbreak, and then pretty much it dies. Okay, so when we get to that workbook lesson, small lines mean small answers, long lines mean sentences. Okay, now let's go to our review for the quiz. Now, in the lysogenic, a lot of times the host cell keeps on replicating virus DNA, but anytime it can become lytic and explode. So I would say lysogenic, that host cell, I don't even know if I could go back. So look up here, here's a good representation. This is lytic, lit match, explosive. Lysogenic, the host cell just keeps on replicating that viral DNA. So I would say in the lysogenic, the host cell is still alive, replicating viral DNA, but at any time it could go into the lytic cycle, assemble viruses, and then have to release all of them, basically killing the cell. Okay, name on this quiz review. Okay, y'all are gonna answer this for me. Viruses are made up of 2 things. The inside has what? Made from the biomolecule what? So the inside has what? Yeah, say it out loud.
Genetic material.
Ooh, I like that. Genetic material. Or you could have said DNA or RNA. Made from the biomolecule. What is the specific biomolecule that DNA and RNA are? Nucleic acids. Good. So ready? The first fill-in. Viruses are made up of 2 things. The inside has, you could even write genetic material. You could write DNA or RNA. Then the second fill-in, made from the biomolecule, nucleic acids. You have to write those out. And the outside is called the blank. Starts with a C. Good. Some people might even write protein capsid. That's fine. Made from the biomolecule. I just said it. Protein. Okay. Okay, number 2. Viruses are considered, circle it, living or dead? Dead, I agree. Because 2 things. They can't what and they can't what? They can't reproduce, and they don't use there's actually 3 things that our notes went over. The one is they can't reproduce without a host, and then there's 2 more. So there's things that our cells do, but their cells can't,
Feed.
which is called reproduction, right? When you don't eat, you die because we need what? Food, energy. So we ready? Viruses don't need energy. They don't metabolize ATP. They also don't have organelles. They don't have cellular structures. Okay, so those are the 3 things that tell us apart. Don't reproduce, don't need energy, don't have organelles or cellular structures. Okay, number 3. Compared to a plant or animal or a bacteria, a virus is, circle it, bigger or smaller? Smaller. Scientists did not know about viruses existed until, we're going to say, the invention of the electron microscope. The invention of the electron microscope. So we did not know about viruses until the invention of the electron microscope. You won't be tested over that, but it's a little fun fact. Electron microscope. Okay, next one. We are going to circle only the things that apply. Number 4, lytic, lit match. Are we going to say that's an active or an inactive? Y'all, the word inactive, I think, means dormant, chill. So, lytic, lit match, is that active or inactive?
Active.
Agreed. Circle it. Host cell lives or host cell dies? Dies. It's active, it's releasing. Here's that picture, ready? Active, releasing. Okay, so lysogenic, we're gonna say is inactive, which means it's laying dormant. And y'all, what's happening to the host cell? It's living. So for the second one, describe the lysogenic. It's inactive, host cell lives. But I need to remind you, at any time, it could turn into lysogenic. Okay. Let's do 7 through 10 as a quick quiz review also. Number 7. What is the most common threat to a host organism posed by an invading virus? Okay. So, first of all, look up here. What happens to a host cell in one of these cycles? It dies. So don't you think that's the worst thing that could happen? Do any of them say that? A, production of viral fluids in the bloodstream. Is this producing any fluids? Nope. Fermentation of acids in the digestive system. Did we ever talk about that?
No.
Destruction of cells by viral reproduction. Yes, obviously. D, stimulation of muscle tone in the heart. Oh yeah, I'm gonna get a virus and my heart's gonna stimulate muscle tone. No, that don't make sense. Okay. 8, which of the following is found in both cells and viruses? So stop what you're doing. What is 2 biomolecules that are found in both? Let's see if any of them say that. Ribosomes and that make protein, genetic material in the nucleus, digestive cavity which drives viral reproduction, or DNA or RNA as a form of nucleic acid. Absolutely D, whoever yelled that out. So guys, it's protein but not A, ribosomes as protein, and it's not B, genetic material in the form of a nucleus, because viruses don't have a nucleus. Okay, 9, almost done. People infected with the human immunodeficiency virus, HIV, have an increased risk of dying from a secondary infection such as pneumonia. What did we say our primary infection was? The first infection you have is always your primary. So what is a secondary? Good. So in this person's case, people infected with HIV have an increased risk of dying from secondary infections such as pneumonia. So what is this person's primary infection?
HIV.
HIV. What's its secondary infection?
Pneumonia.
Perfect, okay? Which of these best explains how HIV increases the danger of secondary infections? A, HIV produces antigens that damage red blood cells. B, HIV adds genetic material to harmful microbes. C, HIV destroys helper immune system T cells. D, HIV consumes beneficial microbes in the body. Okay, which one sounds like it's hurting the body? Destroys helper immune system. Yes. C. Now guys, we can talk more, like the more we learn about biology, we learn that microbes are bad. So there's no beneficial microbes. HIV does add its genetic material, but not to harmful microbes. So C is the best answer. Okay, number 10, last one. Viruses can be transmitted in a variety of ways. The virus that causes SARS, severe acute respiratory syndrome, can be transmitted when an infection person coughs or sneezes. Ew. The virus is transmitted in a manner most similar to the transmission of. So guys, how did they say you get SARS? When people cough or sneeze. So in this chart here, on the left side, it says the virus, and on the right side, it says how you get it. Which one is the most similar to SARS from a cough and a sneeze? So you either have direct contact, exchange bodily fluids, mosquito vector, deposition of airborne droplets.
I think it's
D.
So, y'all, a cough or a sneeze, what am I doing? Achoo!
Oh yeah. So it's influence.
Y'all, look up here.
Influenza.
Y'all, look up here.
flu.
Okay, direct contact.
Airborne. I'm done.
Yes, okay, so look up here. I think y'all have just never learned about this. Exchanging bodily fluids is usually like if you're kissing somebody, swapping saliva, swapping body fluids. Y'all, coughing or sneezing ain't exchanging body fluids. It's probably deposition of airborne droplets. Ready? If I go achoo and then I feel something on the back of my neck, they're nasty airborne droplets hit me in the back of the neck. So which one is most likely?
Influenza.
Influenza, deposition of airborne droplets. Okay,